Yeah. So I'll have more detail. Last time was supposed to be the more public lecture. This one I have more of the details. And so the things I want to start with is we've learned over the last couple of years, we've learned how G proteins and GPCRs are coupled. And I'll talk about that. And also we learned about the mechanism by which the G protein gets activated when the ligand comes up to the G protein GPCR complex. And then we've also learned about beta-arrestin activation, which is important for some systems. And I'll talk about this mechanism that we've now established for a number of the GPCRs. And I'll talk about some of the activation methods. And some of the studies we're doing haven't yet led to a new drug, but the things we're working on. So against on G protein coupled receptors, a ligand like morphine binds on the outside. It causes a G protein on the inside to open up and release a GDP that gets exchanged for GTP. In the case of morphine, that leads to relieving your pain. It can also do what's called beta-arrestin signaling, and for morphine, that leads to some of the side effects. So we'll be interested in both those, but mostly we're going to focus on the G protein activation. So there are around 834 GPCRs in your body. They're in the membranes. One protein goes to the membrane 7 times. And they pack in the membrane. They're alpha helical, and they pack sort of in a closest pack pattern. And there's experimental structures for around 67 of them now, 25 of which are activated. We've also done about 30 structures from theory that are, I claim, is just as accurate as experiments. And the problem is that almost all the time when we try to look at an application, designing a new drug for GPCR, there's no structure. And so we need to have ways to predict the structures. And it's not just one structure. From crystallography or cryo-EM, there may be a structure for an activated system. That structure may be a little bit different depending on which ligand you're using. And sometimes you want to block activity, so you want an inverse agonist, and so you want a structure that's for an inverse agonist, and that's very different than the activated structure. So I think we probably want about 25 structures for the active state and about 25 structures for the inactive. So the question is, where are we going to get those structures? First, about the signaling. There's 2 kinds of signaling. There's the G protein signaling, and there's the beta-arrestin signaling I just talked about. The G protein signaling is generally pretty fast. It's seconds to minutes. And the beta-arrestin activation is long, maybe minutes to hours. And mostly I'll talk about the G protein signaling today, but we have one very important application where beta-arrestin is involved. Yeah, so this slide was made by one of my students or postdocs. I don't like slides with many parts to them. So anyway, the point is that GPCR signaling works with an agonist binding to the extracellular region, and it can do the G protein activity to get G protein signaling, or it can do beta-arrestin activity to get beta-arrestin signaling. Morphine does both. This relieves the pain. This causes side effects. And so we started working on how to predict G protein coupled receptors in 1998. The first crystal structures for rhodopsin came out around 2002 or 3, and the first one for a real GPCR activation process came out in 2005. We'd already published several structures by then. So in the initial methods, we worked out how to predict which membranes are in the middle, how they interact, how they bind. But these days, now that we already have 67 experimental structures and 30 theoretical structures, what we do is, when we want to get a new structure, a target, we look at the various templates available, and we compare the homology between our template and the ones that are available. And we take several of those structures and use those as templates. Once we have the template, we have the tilts defined. as according to, say, the experimental structure. And then we just rotate the helices about their axes. And what we've shown is that if we rotate every 30 degrees, so 12 different rotations for each of the 7 helices, and evaluate the energy for all 35 million structures, and then build them into 7 helix bundles, the best 1,000 or so. If we start with a known crystal structure, we almost always end up with that as number 1 structure out of the 35 million. And that says that the methods we're using to predict where the side chains are, the methods we use to evaluate the energies are good enough to be able to recognize the winners. There's only 12 pairs of interacting helices. If the tilts are fixed, there's just 12 pairs to worry about. So 12 rotations of each pair of helices, it's only 144 structures. So we only have to do 1,728 pairwise interactions to evaluate the energies of all these 35 million. Now the problem is, when you do that by helix step, each of those pairs of interactions, we optimize the side chains for that pair, but the same helices may be involved in another pair and they'll want different side chains. So this is what's called a mean field. The 35 million is a mean field analysis. And then we take the best 1,000 or 2,000, we build that into a 7 helix bundle, we redo the side chains. We have a very fast procedure for optimizing side chains, this screen procedure. And then, so that gives us, again, we do the best 1,000 structures, and then we look at the best 20 of those 1,000. This is an example. We're not doing the combi helix, just doing the pairwise interactions for rhodopsin. The number 7 structure is actually the crystal structure, but when you build in the 7 helix bundles, it's number 1. In almost every case we've done, the best one's number 1 for the apoprotein. In this case, what you can see is for rhodopsin, there's lots of variations in the rotations of TM5, 6, and 7. These are just 5 of the 30 systems we've done. So doing this combi helix, building the 7 helix bundle, the crystal structure is number 1. Of course, we don't want the structure for the crystal structure, it's already known. We want the structure for the new target. And so the problem there is that if we start with a template of a known X-ray structure and we're trying to predict the structure of a new target, it's going to have a different set of rotations. So this just gives you an example of different GPCRs and the variations in the various tilts. So, I mean, the tilt angle changes by plus or minus about 10 degrees, azimuth angle by plus or minus around 30, and the rotation angle by plus or minus 30. So we need to optimize the tilts and the rotations. So what we found is that if we've already done the bihelix step, and now we're going to do the tilts, we call that super bihelix, that if we do the tilt angle by plus or minus 10 degrees, the azimuth angle by plus or minus 15 and 30, and redo the rotations by plus or minus 15 and 30, we think we sample all of the important configurations. It's 13 trillion, somewhere here, I guess. Yeah, there it is. So if we sample those 13 trillion, that the correct answer is in that 13 trillion. And so, again, we do the pairwise interactions. It's a little more complicated because there's different tilts involved. But we evaluate the energy for all 13 trillion. We take the best 1 or 2,000, build into 7 helix bundles. We re-optimize the side chains using SCREAM. And then we reduce that down to the best 25. And so we think that the best 25 structures involve different kinds of small rotations, different amounts of small tilting, and different ligands may want one or the other of those 25. We think we need to do all 25 to guarantee that we have a starting structure close to what that particular ligand would like. And this just shows you the various little ligand structures. I don't know why I put this in here. Oh, I see. Yeah, this just shows you that that these best 25, these are the best 20 structures, and this is the kinds of variations we have in the various angles. And for adenosine receptor, these are the kinds of variations in the top 10 or so structures. We don't generally keep 25. And for the kappa opioid, these are the best 10 or 15 or so. In all three of these cases, number one structure was actually in a known crystal structure. So what we found is that very often the good agonists bind to a higher energy structure. This case for adenosine, all four of the good agonists bind to number 15. The best antagonists bind to number 2 and 3, and no one binds the very best structure of the apoprotein. So that's why we keep about 25. So those calculations I just told you about, there's no membrane. We had the 7-helix bundle, we had templates we started with, we did all these rotations, evaluate the energies, and there's no membrane yet. So what we do is we then take these best 25 structures, we add on the extracellular, intracellular loops, put it into the membrane, put in the physiological salt and water and stuff like that. So it's around 50 to 100,000 atoms, and we do molecular dynamics. And if we've made a mistake, what we expect is the molecular dynamics, we're going to lose the context we predicted. If we didn't make a mistake, we expect to actually find new interactions that improve the structure. And so, and we can find that out in just a few nanoseconds. So we don't use molecular dynamics to make a prediction. We use molecular dynamics to validate our predictions. Again, there's no crystal structures to compare with, so we need validations. So now we have structures, we need to dock ligands. And so we developed a procedure we call Darwin dock. So we call this procedure we use for predicting the structure of the GPCR, we call it complete sampling. We claim that 13 trillion structures is doing all the relevant structures. For docking, we do the same kind of thing. We take each one of those 25 structures. We also do a conformational sampling of the ligand. And generally, there's various conformations of the ligand. We take the best 10 or 15 of those ligand structures. We dock each one of these 10 or 15 to each one of these 25. And we, for each one of those cases, we do this Darwin dock process. So we take we put spheres in the system where the ligands can go. We put in about 50,000 poses of the ligand without any calculations of energy. We do the RMS differences between those 50,000 structures. We group them into families. There are about 2,000 families. We then do the energy of the family heads interacting with the protein. We take the best 10%, the best 200. We take all the daughters of each one, about 25, and we do the binding energy calculation interacting with the protein. But all those calculations, we ignore the 6 big hydrophobic residues. So it makes plenty of room for this ligand to find good positions. From the 50,000, we reduce it down to 100 best ones. We put back the hydrophobic side chains. But now we use the screen procedure. So each one of those 100 structures, they all have a different best configurations of these hydrophobic residues. So the big problem with predicting binding sites is a chicken-egg problem. We need to know where the side chains are to figure out where the ligand goes. We need to know where the ligand goes to figure out where the side chains are. This is how we solve the chicken-egg problem. We take the hydrophobic residues, phenylalanine, tyrosine, replace them with alanine, do all our docking, get the best 100 structures, then we put back the hydrophobic residues. So we call that process a Darwin dock. And we tested it lots of time. Almost always, if we do it for some cases, a known crystal structure, and we ignore the side chain positions in the crystal structure, we go through this process, we get no more than an RMS deviation of 2 angstroms between our predicted structure and the crystal structure. Very often, maybe more than half the time, we have less than 1 angstrom difference. So we think it's a pretty good procedure. Yeah, so that's the methodology. These are just a few examples. Again, we don't use a crystal structure residue positions. We do it from scratch using Scream. And the various cases are almost always within 2 angstroms. This is 0.3 off. This is 0.8 off, 1.3 off. So it works pretty well. So now, the really important issue with GPCRs is how does it get activated? What's the mechanism? So people know the structure of the inactive for 40 different cases. They know the structure of the active for 25 cases. No one knows the structure from experiment that actually does the activation. All they know is after it's activated, what structure they end up with. What we need to know is the structure that gets activated so we can design ligands to do the activation. So the hint was the first cryo-EM structure for a GPCR that had the G protein was done for mu-opioid receptor. It was missing about 150 amino acids, but it gave us a starting point so we could use our computational techniques to figure out where the rest of the 150 were and get a structure. So we know mechanism for opening up and do the G protein activation. We also want to know the mechanism for doing the beta-arrestin activation. So here's the story. We published this year, 2022, PNAS. So you have the inactive structure, no G protein. Very often the inactive structure on the intracellular end of it has a lock between TM3 and TM6. And then what we find is that what happens first is the G protein has an alpha, beta, gamma unit. The G protein comes up. The G alpha C terminus is sticking out from that G protein. It has a negative charge at the C terminus. That C terminus comes up, the salt bridge breaks the salt bridge. Then the alpha 5 helix of the G alpha, it gets inserted partially into the membrane, but the GDP is not activated. It still has a GDP bound. So you end up with this pre-activated structure, G protein with GDP tightly held between the RAS and helical domains, and the G protein is partially activated, the G protein is partly inserted, and it waits patiently for an agonist to come up and do something. So then the agonist comes up, binds to this pre-coupled complex. That causes the G alpha to open up. It releases the GDP. Actually, the GDP stays with the RAS domain, but it can exchange now for GTP. That does the signaling, relieves your pain, or if it's dopamine, it makes you feel really great. You got into the university you want to get to, you passed your PhD exam or whatever. For experiment, we know these structures. For experiment, we know these structures. No experiments have yet determined these structures. So only know this from the theory. I think with the publication, so we did this for mu-opioid about 2 years ago, submitted a paper to Nature Chemistry. And after a year of deliberating with reviewers, they couldn't find anything wrong with the paper except there was no experimental confirmation. And so then what we did was we did 15 structures and 6 GPCRs, and that was good enough. We got that published. And I think with the publication of this paper, there will be motivation for people to actually find this important structure. This is a structure you want to do drug design. You want to figure out where the agonist binds to this structure to cause a low barrier to open up and do the activation. So here's how we got our start. There was a cryo-EM structure for the mu-opioid receptor. This is what they found. They were missing 138 residues. And this is the final structure when you add those missing residues. So the most important thing is that the crystal structure missed 12 salt bridges between the G protein and the GPCR. They just didn't resolve the salt bridges. The backbone is pretty good, but there's too much disorder to get the salt bridges. And these 12 salt bridges, they couple that G protein to intracellular loop 1, intracellular loop 2, intracellular loop 3, and they hold the G protein at an angle, and that angle has an alpha 5 helix positioned exactly where it needs to go to get activated. So it's a really important result. Mostly, the cryo-EM still have not resolved the salt bridges. So what we did in our calculations was we used the backbone from the cryo-EM, did all the optimization to get with metadynamics to find where all the salt bridges were, and then we let the system go, And we actually got slightly better resolution than the X-ray structure to the experiment. So this just compares the two. This is the structure from the cryo-EM, and this is the structure we add everything. You can really see all these salt bridges that couple the intercellular loop 1, 2, and 3. So I think this is still saying the same thing. I'm just repeating myself, I think. Let's get some more detail about them, but we won't focus on the detail here. So this is just an example of one of the systems. This is the serotonin 2A receptor. And the GQ G protein. And this just shows the energy to break the ionic lock. It's downhill 2.7 kilocalories. This is the final position of that C terminus that opened up the 3,6 ionic lock. It finally ends up bonding to TM6, mixed salt bridge, and this is the energy there. So both these processes are downhill 2 to 3 kilocalories. We get that by doing metadynamics. So we use the GROMACS code, and then the various ways of doing the metadynamics. When you have high barriers like these systems, we do the metadynamics usually about a half a microsecond to get good free energies. So these are the 15 cases we published in PNS earlier this year, showing all 15 cases. It's the same thing. G protein comes in, makes a complex of the GPCR. That complex, when the agonist binds, activates the system to get the product. And here's just a couple of examples of some of the energetics. This is the mu-opioid receptor. Going from the pre-coupled state to the final system is downhill in energy about 6 kilocalories, plus or minus about 0.1. And in that process, you open up the G-alpha, and the GDP now is available for exchange. There's another case. This is serotonin 2A receptor. And what we find here is that the full agonists have a small barrier and a big delta G. Partial agonists have a big barrier and a small delta G. People argued about what's the difference between a partial agonist and a full agonist. Well, the difference is that in the full agonists, they all go to the activated structure. In the partial agonist, part of the population is stuck here, part of the population is stuck here, so you don't get the full effect. And then inverse agonist, it's way uphill to activate. So the inverse agonist prevents, bound to the G protein GPCR complex, the inverse agonist prevents activation. So this is another comparison, another ligand. This is just comparing the final structure. So the final activated structure that we calculate and the final activated structure measured experimentally are in excellent agreement with each other. So this is for beta-2 adrenergic receptor. This is the one you want to agonize for asthma. Again, it just shows that the process of going from this pre-coupled structure where the G-alpha was tightly holding on to the GDP to the final structure, downhill 16 kilocalories in this case. Again, a comparison between the crystal structure, fully activated crystal structure, and our predicted structure, excellent agreement. So here's the overall picture. You start off with the inactive GPCR. It generally has an ionic lock that holds it inactive. G protein comes in. The C terminus of the G-alpha breaks that ionic lock. It gets partially inserted and waits patiently with the GDP still held tightly until the ligand comes in. The ligand comes in, activates it, and then you get this full opening up of the G-alpha and releasing the GDP. So that's the mechanism. Again, all 15 of these cases for Class A works that way. Yes, go ahead. And by the way, I'll just keep talking without interruptions unless someone says something. So go ahead and ask questions. We have time. Yes, Prabal.
So this one, previous slide, the intermediate.
Second.
This was in a membrane.
All these calculations, what I'm talking about here, they're all in membrane. Well, we do the predictions outside the membrane. We take each one of those 25 structures, we add in the intracellular, extracellular helices, We insert them in the membrane with physiological salt, generally for a class A, about 50,000, maybe 75,000 atoms. We do the dynamics, generally at least 10 nanoseconds, usually 100 nanoseconds.
So these are like ligand-bound form, ligand-unbound form, correct?
Yeah, so this is the inactive, no ligand, no G protein. This is bringing in the G protein. It binds the system. It goes downhill to open up that ionic lock, and then it relaxes to configuration. It's partly open. So I think the 10 angstroms here is so it goes from 10 angstroms to about 15 angstroms. It finally gets activated. It goes up to 21 angstroms between the.
These are like 4 different set of simulation.
Oh yeah, yeah. So we start here. We actually bring in the inactive G protein, GPCR, the inactive G protein. We let them interact, and that C terminus interacts with the ionic lock, the salt bridge between R350 and E whatever, and opens up the salt bridge, and then it relaxes with 10 angstrom separation to this final separation of 15 angstroms, still keeping the RAS domain and the helical domain of the G alpha holding on to the GDP tightly. Only when the ligand comes in does that open up. So where the ligand binds is very important, because that determines whether it's going to be a full agonist, a partial agonist, or an inverse agonist. And that opens up to the final structure. These are all in a membrane. Any other questions? Yes?
When you selected those 25 best morphologies,
One more time.
when you chose the best 25 morphologies from the trillions of them, so was it just the energy parameter or something else?
Yeah, we use energy.
Just the total potential energy.
Yeah, so
Okay.
the only criterion we have to make choices is energy. So it's too bad, the force fields, amber and charm, they're best currently force fields that are practical for proteins. They have crappy non-bond interactions, they have crappy electrostatic interactions, and so it's not so reliable. When we do our structure prediction in Ensemble, we use a different force field called Dryden that has very accurate hydrogen bonds, and that's really important, we think. So the ensemble calculations that get the structure, the Darmstadt calculations to pick the binding, that's all done with Dryden with very accurate hydrogen bonds. But then we put it in the membrane, and then we're stuck with charm and amber.
Right.
And right now, they're not great, but they're good enough to be okay. We hope to improve them over the next few months, get much more accurate non-bonds, much more accurate electrostatics for them. But right now, this is what we use. So they've been optimized. The charm and amber have really developed a lot in the last 30 years. They're actually not too bad now. So last year, up until we published this paper, people believed that the way these things work was you had the inactive GPCR, the ligand came in, and some kind of way opened up the inside part of the system to recruit the G protein to get activated. So this is a general Kobielko model for how G protein work. So we've proved this is wrong from a number of cases. This is one of the examples. Here we took the serotonin 2A with the final activated structure. The G protein's bound, the ligand's bound. Then we remove the G protein. And what happens is it goes from the active structure back to the inactive structure. That agonist cannot keep it in the activated structure unless the G protein is there. And it's pretty big energy, 6 to 8 kilocalories. These energies are all from metadynamics, and the uncertainties here are generally around 0.1. Well, they're quoted here, 0.3, 0.1. So this is beta-2 adrenergic receptor. This is showing that starting with the inactive form and binding the ligand, it can't open up the ionic lock. It's uphill 15 kilocalories to open up that ionic lock. So it can't do it. Without the G protein, no activation. And this is the mu-opioid receptor showing that the full agonist cannot open it up. It's uphill 13 kilocalories to go to the activated state. So without the G protein, no activation. Without the agonist, the G protein already partially activates the system to get a pre-coupled state, but the G protein is not activated yet. It's holding in place, waiting for the agonist. So that's what this says. So, all right. So now I want to talk about, so that's the mechanism. I just want to talk about some of the cases we're working on. So we did a project with NIH. So our experimental collaborators found that binding this protein with 450 amino acids to serotonin 1E. 1E is it's a serotonin receptor, but no one had any idea what it did. There was no known function for it. And so, what they thought was it caused beta-arrestin activation, and that mediates stress-induced neuroprotection. So this is potentially, if you could replace this 450 amino acid protein with a nice small ligand, you'd have a really important drug. So in this case, let's see what I have. Here's a paper. It was published this year. And so what we want to do is, so we already found the binding site. I'll talk about it in a moment. And then we're going to there's the binding site where the protein binds to this serotonin. So it's all in this extracellular loops here. And what we want to do is take that binding site, get a pharmacophore, do virtual screening, and find new small molecules that could be good for beta-arrestin activation. This is being worked on by a senior at Caltech. So again, I'll just go through the procedure we use to get the structure. These are the templates we use to predict the structure. They're all serotonin derivatives. They're all X-ray structures in all 6. We used all 6 of them. We do all 6 of these structures with this bihelix form, and then we compare the 35 million structures from each of the 6 to find the best 25 total. And these are those 25. They mostly come from 2 or 3 of the systems here, 2 or 3 of the starting points, the templates. And we notice here, so there's a criterion. We did a paper a couple of years ago analyzing what contexts are common to GPCRs that are activated, what contexts are common to GPCRs that are not activated. So when we look at a structure, we can get a hint as to what it's likely to be active or not. And these structures here, there are 7 or 8 interactions associated with being active. So we used these structures here. We used these 4 shaded ones. That was from bihelix. So the bihelix, remember, we go 35 million structures. We boil it down to the best 1,000. We did this for 4 cases. We had 4,000 structures. We take the best 25 of all those 4,000 structures, and that's what these are. So then we did super bihelix. So here we took 2 templates. I only circled 1. We did a second one also. We do the 13 trillion structures. For each of those 13 trillion, for those 2 templates, we take the best 1,000, we merge them together, take the best 25 structures, and this, oh, I see, yeah, okay, so, yeah, so, anyway, so this is the best 25. Then we picked 2 of these structures. Oh, okay, this is just showing this is showing the sequence alignment of serotonin 1E to the other serotonin receptors. And all of these targets we were matching to, I mean, they're experimental structures, but they got missing residues. So, I mean, it's nice to have the structure, but you need the residues also. So we added these 144 residues. We either use MD or meta-MD to get the full 3-dimensional structures. And so here's our final best structure for serotonin 1E. It's got a 1,2,7 coupling, a 2,3,4 coupling, some special things that indicate that it's activated. Then we do, in this case, we did 100 nanoseconds. We first did restricted structures. We restricted the backbone, did some dynamics for about 5 nanoseconds. We relaxed those at another 5 nanoseconds. Then finally, we did fully relaxed to show that it was converged. And then we finally did 100 nanoseconds to get the final structure. And this shows the active site. for the serotonin receptor after we finish. There's a lot of interaction between the extracellular loops and this protein. The experiments had shown that only of this 450 residues of the protein, the CPE protein, 150 to 300 was sufficient to do the beta-arrestin activation. We did these 150 residues, and these are the salt bridges we find. So what we want to do now, find small ligands that can match those. So yeah, so then, and we did 1.5 microseconds of microdynamics on the CPE protein bound to the serotonin 1E. And this is just showing, again, the best interactions we got from that. And so Ellen Kang is a senior. She's now taken these various interactions we identified that CPE recognizes serotonin 1E. It does the beta-arrestin activation. We've also shown, I have a slide on it, that structure does activate beta-arrestin. And now what Ellen's doing is she's going through doing in silico screening. So we use the phase program and Schrödinger's tools to do the in silico screening. I don't know how big, we may be doing 160 million. I'm not sure what size she's dealing with right now. That's pretty extensive. It takes about a month to go through the 160 million with this active site. So I think we may be doing a subset. But anyway, what we want to do is get a small ligand that can replace it. So yeah, this is just showing, again, the active CPE protein bound to the GPCR. So we took that structure. After we had the CPE bound to the GPCR, then we phosphorylated, took off the G protein and phosphorylated the intracellular serines and threonines because that's done by an enzyme. Then after doing that, the beta-arrestin can come in and interact with the intracellular region and either get activated or not activated. What we found in this case is that it activated the serotonin. It led to activation. So that's one project. We're hoping that, so Ellen's a senior this year, we're hoping before she finishes, she'll have identified some novel ligands. The 160 million in the PubChem database can all be ordered in principle. Most of them come from Russia, but anyway, they can be ordered. And then we have a collaborator at University of Arizona that can measure directly the receptor ligand interaction. So she'll test our best 5 or 6. So hopefully we'll find a hit. If we do find a hit, NIH is ready to go do the experiments. So the other project we've been working on is substance abuse. So there's an experiment showing that if you could agonize an orphan receptor, GPR139, no one knows what it does. At the same time, if you can block or at least not activate the D2 or maybe D3 dopamine receptors, it's not clear whether you want to block both of these or just D3. But what happens with mice is that cures substance abuse. They get mice that are hooked on alcohol, and then when they take ligands that can agonize GPCR139, and they block the dopamine receptor, 2 different ligands, they can cure substance abuse. And so we want to get new drugs that we want one drug that does both. It interacts and agonizes 139. At the same time, it blocks D3. So again, so these are known agonists for the 139. This is the one I like best. There are 2 of these that we've done docking to. So again, we went through the bihelix procedure. We picked out 4 cases that use different templates. We used those to do the super bihelix. We focused on these that have, this one has 4 active sites. We did the super bihelix. We pulled out 6 cases here of the super bihelix that we then are binding the ligands for GPCR139. We then generate the loops on it, put it in the membrane so we can do dynamics. And so, we've done this now for about several known agonists. And so now what we're doing is, let's see. Okay, so someone else did high throughput screening to find 36 agonists. And so, we're looking now at these 36 and other ones that come out of our in silico screening to find the ones that could also block D2 or D3. In the cases we find that can do both, agonize GPR139 and block D2 or D3, Judy Sue at Arizona, she'll measure receptor binding for both those. So those are the two activities. I'm hoping we'll have small molecules identified over the next few months. So next time I'm in India, maybe we'll have solutions to them. So we also have several projects in Class C GPCRs. So the dopamine receptors, the mu-opioid receptors, the GPR139, they're all Class A. They have one transmembrane region. But Class C has two transmembrane regions coupled together. And they tend to be pretty complicated. This shows a typical example. This is the sweet taste receptor. So this is a receptor in your tongue that recognizes sucrose. And so there's a pair of them. They're identical. It's a homodimer. The G protein binds to one of these ends, and the sucrose binds first. In this case, the sucrose binds first. The G protein comes up, and the sucrose is already there. It falls down and gets activated. So we've done the simulations on that. Okay, well, I'm sorry. So Class C receptors we've been looking at are GABA. They play an important role in neurotransmission, and these sweet receptors. And so, yeah. Okay, I guess that's the story. This is a sweet receptor. And then for the GABA, there's a cryo-EM structure. It was almost complete. So we've done the full simulations on starting with that structure. The G protein binds differently with the GABA heterodimer than it binds to the sweet receptor. And now we're trying to determine the mechanism. We haven't proved it yet, but we're pretty sure the GABA receptor, the G protein binds first, and once it's bound, the GABA then can bind to the extracellular region and call activation, but we've not shown that yet. This is just showing the couplings in the system. Again, we get these couplings. Generally, the cryo-EM doesn't see the salt bridges coupling the intracellular region. They basically, those residues are just this order right where the salt bridge would be. Yeah, and this is comparing where the alpha 5 chains are in these different receptors. So the GABA receptor, the Class A, I've forgotten what receptor this is. Anyway, so these chains are different in different GPCRs. In the case of a GB2, the active and inactive, the helices are very similar, but they're very different in these others. So we're just trying to figure out what happens in those systems. This is showing how the G alpha unit gets activated from the inactive GPCR, the GABA GPCR, as the G protein comes in. The G alpha opens up and they get the exposed. So we think this is the key step, opening up the G alpha to expose the GDP. So this is the overall mechanism for the GABA receptor. You have an inactive structure. It has a 5-5 coupling between the helices. And then as you go towards activation, you get a 5-6 coupling. And then finally you get a 6-6 coupling. So, and then in the process, the agonist gets bound here, and then goes from the 5-6 to 6-6. So there's some details on some of these structures now from experiment. We're doing it all with the simulations. So again, there's structures for the activated, the structure inactive. There's not too much known about these intermediate structures. So let's see. I think I was going to talk about some detail about the movements. Still a lot of questions we're asking. Yeah, again, we found so far, we've shown that for the GABA receptor, that if you put the agonist and do not put the G protein, that it does not get activated. So we've not yet proved that if you put the G protein in, you get a precoupled structure and then the agonist activates it. Yeah, so the metabolic glutamates are also class C. They involve dimers. And they have a structure that does not have a ligand. It has a G protein coupled GPCR. We think that's evidence that the G protein binds the GPCR first. So again, we go from inactive, the G protein binds, we get the precoupled structure, agonist comes in, activates. We've not proved this yet, but we're pretty close. This is the sweet receptor again. This is a homodimer. Sucrose binds in these sites out here. Now this is where the transmembrane region is, down here. This is a transmembrane domain. And then there's what's called a cysteine-rich domain, and then there's what's called a venous flytrap domain. And here's down the G protein down here. Mechanism is sucrose bind here, and then around 6 to 8 nanometers away, the G protein opens up and releases GDP. 6 or 7 nanometer separation. It's amazing. So we can simulate it. We don't fully understand all the interactions that are going on. It looks like binding to sucrose affects the cysteine-rich domain, and that affects the top end of the transmembrane domain, and that affects the G protein. However, we've been able to simulate the process. As you go from inactive, no G protein, the G protein comes up, it starts interacting with the sucrose, it goes through a state we call S1, goes to another state we call S2, and then the extracellular region, you had dark blue on the left and pink on the right. Over here, now you have, in the extracellular region, dark blue's on the right and pink's on the left. So you twist about 100 degrees as you go from the inactive to the activated structure. The last step of activation, we get this 100-degree twisting going on. And so we thought this was pretty exciting. And we submitted a paper to JAX reporting it. And reviewers had a lot of complaints. They wanted longer dynamics and all these sorts of things. We answered all those questions. They still weren't happy because there was no experimental evidence showing that it worked this way. That is, that you had the sucrose and the G protein came in, caused this twisting motion. So it got rejected. And while it was being reviewed, 4 Nature papers came out reporting, I think, 8 or 10 cases of metabolic glutamate receptor, and all 10 of them did exactly the same thing. It rotated 100 degrees as you went from the inactive to the activated. So I wrote back to the editor and pointed out these papers that had come out while it was being reviewed, and it got accepted, so it's got published. So let's see. Yeah, so this is just talking more about the details of going through these intermediate states. Back here, I talked about S1, S2, S3. And this is just looking at what's happening in these states in terms of how the GDP is interacting with the RAS and helical domains. You open it up, there's some water exposed, and finally you get the whole thing open. And this is where the twisting occurs. So I think, so we've also looked at the heterodimer. So our funding for doing a Class C came from a food company called Cargill. Cargill wants to market sweeteners in Europe. Europe requires sweeteners be natural. You cannot do artificial. And so there's a plant in Chile, blanking on the name now. Anyway, there's a plant in Chile that has sweet. It's sweet, but it leaves a bitter aftertaste. And so Cargill funded us to figure out how to get sweeteners that can be derived from this plant, so they're natural, but do not cause a bitter aftertaste, which comes from binding to taste receptors. And so it's the same kind of structure as a homodimer. So I probably had a point to make about this. Okay, so where we are now is we sort of have a plan that's evolved for how to make progress in getting ligands for G protein coupled receptors. We first predict the structure. We use this ensemble technique. We do the bihelix, super bihelix, get the best 25 structures. We take some known ligand, if there is one. We bind it using this Darwin dock procedure to these 27 structures. And then from the docking of this ligand, we get a pharmacophore. So that's, salt bridge here, hydrogen bond there, aromatic hits here, aliphatic there. Usually it'll be about 6 sites in the pharmacophore. Then we can go through up to 160 million ligands to discover ligands that might like that site. So that's how we're doing the serotonin 1E problem. We want to find new small ligands that can do the same thing as 450 amino acid CPE does. But these molecules can be purchased. So then we have a collaborator at Judy Sue who's developed a very sensitive technique of doing immobilizing a receptor and looking directly at the ligand binding. So she can test our predictions. So we go through the 160 million from the in silico screening using the Schrodinger phase program. We take the best 1,000 or 2,000. We do Darwin dock to get accurate energies. And then we reduce that down to best 5 or 10 for Judy Sue to do experiments. If it passes her experiments, then we send it to Steve Liggett at the University of South Florida. He does cell assays. And if it passes his test, they come back to us. We take this ligand, which generally won't be much better than 1 micromolar binding, and now we change we take the scaffold, but now where there's a methyl, we may replace it with methoxy or a sulfonyl group. Do maybe several different sites around, maybe 50 to 100 to 200 various R groups, and then we estimate the energies with Darwin dock, and then we get the best 5 of those. Now we can't purchase these molecules because we modified them. So Barry Stoltz at Caltech has one of his graduate students make them. He makes enough for Judy Sue to do her experiments, passes her experiment, goes to Steve Liggett to do his experiments, passes his experiments, come back to us, and if it's not good enough, we want maybe 1 nanomolar, we go through another cycle of doing R group screening, et cetera. And so that's the plan. We had a proposal in NIH, didn't get funded, so we'll go try again for next year. We're doing it anyway without funding. So Judy Sue's experiments, she immobilizes on the system. She got her PhD at Caltech, but she's an applied physics faculty member at the University of Arizona. So she can measure down to picomolar and nanomolar, atomolar binding. So these are people who did most of the work I talked about today. So Suk Kyung is head of the bio group, bio part of my group. Moon-Young Yang is doing did the GPCR stuff and the taste receptors they didn't talk about. Mir Mafi did the G protein pre-coupled state, those 15 structures, and some of the other things. So I went on. I think we still have time for discussion and questions. So I can go back. I went kind of fast. But we're really very excited. We think we have really good ways of getting these structures. It's really important. Homology methods do not work for GPCRs. If you take a known structure and you do the mutations, there'll always be a bad contact, so some helix rotates. Helix rotates, the active site changes, so you just can't use homology. And so that's what people do in the literature. It's really terrible. They publish papers, but they're really crappy structures. We think we get good quality structures. Now we're to position, what we need to do is develop some good drugs. We want to get to the point we can get some brand new drugs for these GPCRs for the substance abuse, pain without side effects, and so the neuroprotection. So we're on our way, but we don't have Tuesday's lecture I had examples where we've actually done better than the best current drugs, but I don't have examples here yet. Anyway, that's the story. Very exciting.
Questions, comments?
I think we have half an hour.
So I have a question. So how the delta G was calculated? It was by using the enthalpy and entropy, or using any programs like MMPBSA or MMGBSA?
How the delta G was calculated, the free energy change?
Oh, so most of these cases I did metadynamics. So metadynamics you use when you have high barriers to go over. And you can't, they never go over metadynamics, they're too high. So metadynamics, for example, opening up the G protein. We have a closed state where the GDP is held between the RAS domain and the helical domain, and there's two salt bridges that go across that. And when it opens up, you break those salt bridges, they might actually make internal salt bridges. And so we do dynamics where there are two variables, the distance between the salt bridges that go across the GDP, and we let that go from a short distance, a few angstroms, all the way to when it's fully open, it may be 20 angstroms. And then we also do the center of mass of the RAS domain and the center of mass of the helical domain. We let that go from the distance it is when it's not activated to the distance it is when it's activated. And so then, in metadynamics, what you do is every so often, say every 50 femtoseconds, you add a little Gaussian to the energy of whatever it is in that process. And then 15 femtoseconds later, you add another one. And then in the process of adding these Gaussians, you're raising up the bottom so you can sample the transition state more often. And so that process still takes maybe half a microsecond to sample back and forth between the closed and open form to get good accuracy in the barriers. So we generally do it till we get an accuracy of somewhere around 0.1 to 0.3 kilocalories. So that's the process. Metadynamics use almost all of these free energy calculations. It's, of course, no better than a force field. Force fields are not great. We think they're good enough to use for the drug design. It's just that right now, it's I wish we had more accuracy. But it's still, they're good enough. The current force fields, the charm number, are okay. They're not great, but they're okay.
Any further question, comments?
Way back there. But yell out, my hearing, and help, maybe for all you can translate it. Yeah, he needs a microphone.
Yeah. Oh, you have there, okay.
Okay, I think now.
Okay.
Now it works. Yeah. Okay. Thank you very much. That was very impressive. I'm wondering, when you do this complete sampling, what is the main advantage over doing Monte Carlo?
Well, the problem with Monte
Are
Carlo
there
is
really parts? Do you get, if you did Monte Carlo, would you get stuck in some part?
well,
of
the problem with
space?
Monte Carlo. Let's just say we have 400 amino acids, and we want to do 3 conformations for each the 400 amino acids. That's 3 to the 400th. No one's ever going to sample that many structures. So a real problem with Monte Carlo is making it complete, that you're sampling all the relevant configurations. And so the simple-minded way is doing Monte Carlo for protein structures is that you have at least the possibility of an alpha helical structure or a beta sheet structure or a loop structure, just 3. 3 to the 400th is an impossible number. And so the problem with Monte Carlo is making it complete. The problem with vector dynamics is you never go over the barriers. So you get stuck wherever you started. And so we claim that doing the 75 to the 7th structures, the 13 trillion, we claim that includes all the possible structures. All we have to do is identify them. And we claim that using these energies are good enough that we can do really a mean field analysis. We do the helical interactions to get the interactions where the side chains are optimized just for the pairs of helices. We evaluate the 13 trillion, and then we take the best 1,000 or 2,000. We build into a 7-helix bundle. We reassign the side chains using SCREAM, get the optimum side chains, do a little bit of minimization, and we think that then doing those 1,000, we think we're comparing all the relevant structures. So we think we're throwing away a lot of data. I mean, probably there are machine learning strategies we could use that could use the 13 trillion pieces of information and get useful things out of it that would help us. But, so we think that this is a way to sample the complete space. That is, it's practical to do the 13 trillion because it involves a pairwise interaction. It's a little more complicated because the tilts change, but so we worked out a procedure for that. So we think that we get respectable enough energies from the mean field part, the 13 trillion, that then we can trust the 1,000 or 2,000 7-helix bundles we get, and then we redo the side chains 7-helix bundles, and then we think we've demonstrated that if we do this for a known structure, then known structures are almost always number one of that. So that says our energies are good enough. They may not be great, but they're good enough. And the side chain predictions are good enough. They may not be perfect, but they're good enough to actually get the answers. So I think, so problem with Monte Carlo is sampling all the configurations, because if you do it a straightforward way, there's too many. And so the way we're doing it, we think we keep it to a manageable level. 13 trillion is a whole lot smaller than 3 to the 400th.
Any further question, comments?
Yes.
Yeah. Hi. So, is there any chance that if you do the same structure a few more times, you will get a different energy, or like?
Okay, so again, our procedure is we go through the 13 trillion, we then reduce it down to 1 or 2,000, reduce that down to 25. Those 25 will have slightly different tilts, slightly different helical rotations. We think it's very important to have those 25 because different ligands may like different ones, depending on exactly how the ligand wants to bind. And so we think that 25 is a good number. Our docking procedure basically maintains the conformation. So we don't try to change the conformation of the ligand while we're docking. We use Maestro to do a calculation of all the various states of the ligand. We pull out the best 10 or 15. Each one of those 10 or 15 we dock separately to each one of the 25. We consider that's pretty complete sampling. And then, since it's the same residues and the same ligand, we can compare all those energies. So we can compare the best 1,000 structures, or the best 100 for the docking, for each one of the 15 times 25. You take the best 25 out of that. Very often, it's just 1 or 2. I mean, so different configurations and different ones the receptors would be involved. And so those 25, generally we reduce it down to a smaller number, maybe 2 or 3 or 5 or 6. We put those 6 into the membrane with the physiological salt, and we do dynamics for at least 10 nanoseconds to see if it's stable. It's almost always stable. The only time it failed badly was the very first time we did it. And the guy had made a mistake with the charge assignments. So that's the procedure. We think it's pretty complete. We're trying to make sure we want to sample a big space and get it down and make sure we've got all the good cases. So we want to do drug design. We don't want our experimental collaborator to do 1,000 cases or even 100 cases. We want to reduce it down to the best 5 or so. So it's practical to do the experiments. That's when you really win. If you can do the theory on 160 million down to 1,000, 1,000 down to 50, 50 down to 5, and do 5 experiments, that's a big win. And we think we can do that.
Okay.
We have plenty of time, yes.
Recently, the experimental people are starting to look at the cryo-EM structure
Yes.
of somehow the artificial system
Yeah.
with the small amount of lipid molecules, bearing like a cloud. I mean, small nanodisk-like lipid system.
Yeah, so, so these GPCRs can be put in small membranes. And so Judy Sue does that to do those experiments.
Yeah, so I like to ask that how those methods are reliable. I mean, from the theoretical view that
So cryo-EM is good. It's very low temperature, so that might bias us just a little bit. But almost always in the structures we've compared, the most important interactions are disordered, so that you don't see these important salt bridges. The overall structure is fine, but maybe. Again, maybe 150 Kelvin or so it could be the structural changes that occur. But they've been successful looking at G protein, GPCR coupled systems, and that's really invaluable. And it was really important to us to get our first structure with a G protein coupled to have that cryo-EM structure, because we then could find the missing 140 residues and find their structures, and that's when we discovered the salt bridges. The cryo-EM did not resolve them. It had the overall backbone right, but it did not resolve the salt bridges. And so I think cryo-EM is good, and for the GABA receptor, the jury's still out. Cryo-EM's been done, and there's different kinds of different cryo-EM structures have slightly different structures. We're trying to work out which ones are really right. With the GABA, it's a heterodimer. It's very interesting that in a heterodimer, you cannot bind the G protein to the intracellular loop 3. It's too close to where the intersection is. It only binds intracellular loops 1 and 2. Lots of things left to do here. But I think we're on a track where we can trust the results well enough to actually predict ligands that people can measure experimentally. This is not a game for theorists alone. The success are going to be done by doing what theory can do well and doing what experiment can do well. And they're complementary. We can do 160 million ligands. We did a project, I left it out here, did a project on the pain receptor, a mu-opioid receptor, with a colleague at GIST in Korea. And he had an idea of a really novel fold. He had 68,000 structures he claimed he could make. He could make each one of them in 1 to 6 months. But I mean, you can't do 68,000 that way. We did all 68,000, reduced it down to the 5 best ones. And out of the 5 best ones, it turned out he could only make 1 of them. And it was fair, not great. We did a second round where we took the one that was best, did our group screening, and then predicted 5 more for him to do, and one of those was good. And we did a third round of our group screening, and we had 20 cases that he validated, all worked, and they were all biased. None of them had the side effects from morphine. And so that kind of process, the experiments to synthesize the molecules, it may take a month or so to synthesize the molecules. So the theory has to be done to get the 160 million down to 5 or 10 to make practical for the experiments. I think our methods are good enough. We're going to try to get better energies. We're working on better non-bond interactions, better electrostatic interactions. That should make it more reliable, but already it's good enough, we think. So.
Okay. Oh, you have, okay, last question?
Yeah, no, it's not the last question. We have plenty of time.
So on the final structure we got from 30 millions to 1,000, and then we got 25. Can we use...
Say again?
So from the 13 trillion, you finally 1,000, then 25 structure you got.
Oh yeah, okay.
So to get better statistics, can we use the DFT optimization on those 25 structures?
After you got the 25 structure, can we use DFT optimization on those 25?
Well, these are generally too big for DFT. What we're trying to do, so as I said earlier, the force fields are okay, they're not great. The non-bond interactions in CHARM and AMBER are okay, they're not great. Electrostatic interactions in CHARM are okay, they're not so good in AMBER, but they're not great. But that's what we're using. And so what we're doing right now is taking cases where there's some co-crystals, same protein, but several different ligands, and reducing it down to the quantum mechanics on all the residues within about 5 angstroms of ligand. That's about 400 atoms. 400 atoms is pushing it. You can do the quantum mechanics, but it takes a while. And so what we've done is we've taken the residues that are involved, blocked them at the C alpha position and with the angle there, and then we minimized the charm. It's a little bit better in amber, and then we did minimize that with B3OYP quantum mechanics, MO62X quantum mechanics, and 2 versions of Head-Gordon quantum mechanics. We find all 4 systems in the quantum mechanics go from around 80 kilocalories of binding from B3OYP to about 90 for the best Head-Gordon, and they have similar differences. We think now those quantum mechanics numbers, we can trust those for the non-bond interactions, but the question is, how much that bind is due to electrostatics and how much is due to non-bond? And so then we developed what we think is a very accurate way of doing electrostatics we call PQEQ, where we optimize polarization and charge transfer for not point charges, but for Gaussian-shaped charges in each atom. We think that works pretty well. And we've shown that for these 4 cases I talked about, it gives pretty good systematics as you go through these 4 systems. So now we're taking that and then looking at the total, pulling out the non-bond interaction. We're going to try to get more accurate non-bond interactions. We know that the well depths of these non-bond interactions that are used in amber and charm are probably only about half as deep as they should be. And so we're hoping that sometime this fall we'll have a new version of non-bond interactions that we can bolt onto amber and charm doing dynamics. With the electrostatics, we think it may not be practical to do PQEQ with its Gaussian-shaped charges for half a microsecond. So what we're going to do is use PQEQ to estimate the charges on every atom. We're not going to use the rules that they have in charm and amber. We're going to get the charges from doing a PQEQ on the whole protein. That's practical. And then with point charges, we have to exclude the 1, 2, and 1, 3 interactions. With Gaussian charges, we include the shielding correctly, but with point charges, you have to exclude 1, 2, and 1, 3. We've shown now that doing that, for these 4 cases I talked about, those charges from PQEQ now used in the amber and charm, and excluding 1, 2, and 1, 3, we get the same, basically the same electrostatics. So now I think we're getting close to making a final decision on non-bond interactions. We're doing that 3 different ways. One way is a set of non-bond interactions that we developed by doing molecular crystals at the PBD3 level. We showed some molecular crystals of hydrogen, graphite, O2, S8, things like that. We've shown that if we do the PBED3 level, we get the equation of state accurate up to about 100 gigapascal, which says that the non-bond interactions are pretty well using PBED3 with the Grima correction. So we have that set of non-bond interactions. Grima has a set of non-bond interactions for carbon, nitrogen, oxygen, hydrogen, and some of the others that may be okay. And 2 people in a group have done, there's a data set of pairwise interactions of molecules, like residues interacting, done at the couple cluster, singles and doubles plus triples level, that may be accurate. So between those 3, we're going to settle on a new set of non-bond interactions for carbon, nitrogen, oxygen, hydrogen, maybe sulfur and phosphorus. So one of those 3, we're hoping will work out to match these calculations we're doing. We're hoping that if we do at that level, if we can get accurate energetics, we can automate this whole process of going through all these procedures I talked about. Right now, when we're doing the Darwin dock, we get it down to 100 structures. We look at those 100 structures and think about them. We try to order them by electrostatics and by non-bond, but we have to think. We'd like to get it so we get reliable answers without having to think too much. But we're not quite there yet.